All right, so let's wrap up talking about object recognition and vision in the brain with a specific focus on this question about how things are represented in the visual brain that are not faces, bodies, scenes, or words. And a simpler way to think about this question is just to say, like, okay, well, if all these color-coded regions are responsible for things like faces, bodies, and scenes, what's going on here? This is a part of the visual system. What's going on there? What's going on here? It doesn't seem like it's selected for faces, bodies, or scenes. What about there, 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 there? What about things like, oh gosh, what are these things called? Binder clips? What about a binder clip? What about a glass? What about a now topically very relevant face mask? How are these things represented in the brain? And the answer is they are represented not in discrete category selective regions like the FFA or the PPA. They're represented by a thing that's called population coding. So let me give you an example. Here, we're going to now visualize how often, how many times a second a neuron is firing. And here, we're looking at 61 different individual neurons, okay? And what we're seeing here is from these 61 neurons that we're looking at, this is how they all respond when you see an image of this little cross here, okay? And for a point of comparison, this is what those same neurons look like, the same 61 neurons when you're looking at a picture of a triangle, okay? And as you can see, there's some differences in the patterns. Yes, there's some similarities, but there's also some important differences. And here's a simple way that I can make those differences really clear. Look at the pattern of activity here. What do you think you would be looking at in this case? Okay, so just look at the pattern down here and compare it to this pattern and compare it to this pattern. Which one does it seem more likely that you're looking at? Hopefully, what you can see, if I've done a good job of this, is that this is an instance where you're looking at another picture of a cross, okay? So in this case, what I'm trying to get across to you is that it's the pattern of activity that is what differentiates one object from another object. And that for those types of objects that don't have discrete category selective regions, it's the overall ebb and flow of activity, the rises and falls, the highs and lows, that distinguish one thing from another, that distinguish a triangle from a cross, from a cup, from a mask, from a whatever. And so we can actually see this quite easily with fMRI in humans. So for example, let's look at this idea of trying to classify what you're looking at from the patterns. So imagine you're doing an experiment where you have someone look at pictures of faces and pictures of houses, and this is the pattern of activity outside the FFA and PPA for faces. This is the pattern of activity outside the FFA and PPA for houses, okay? If I were to ask you, what is this person looking at here? My guess is pretty quickly after looking at it for a second, even though none of these patterns are identical, you can tell that, oh, this is probably also a pattern associated with a picture of a face, not a picture of a house. And so the idea is that each category has a unique pattern in the brain, not just for the good old-fashioned ones like faces and houses, but also for things like chairs or for things like shoes, where what you can do is you can actually look and see like, oh, there are differentiable patterns of activity associated with each of these different categories outside of those category selective regions. And so what cognitive neuroscientists often do actually is they actually will literally read your mind by looking at a pattern of data in your brain to say and to decode what it is you're looking at. So the way that something like this works is you will initially have someone look at pictures of faces, houses, chairs, shoes, or whatever, and you'll train an algorithm, a pattern recognizer, to understand, okay, this is a pattern is associated with a face, this pattern is associated with a chair, and so on and so forth. And then what you'll do is you will test that pattern recognizer algorithm, and you'll say, okay, here's a new pattern. What pattern is this associated with? Take a second, see if you can figure it out. Clearly not the faces, because the faces are so blue. And what you will do is you'll say, okay, pattern recognizer, look at that pattern, make a guess. And the algorithm will say, I think it's a house. And in this particular case, sure enough, it is a house. And so you'd be like, yes, the algorithm did a good job. It got that correct. It actually turns out that neuroscientists can get stuff like this correct like 95% of the time. Like it's actually almost trivial at this point that you can read someone's mind and decode what it is they're looking at without ever seeing what they look at, just by looking at patterns of activity in the brain, okay? So this sort of idea about pattern activity is how the brain can represent objects that don't have dedicated regions or modules, such as the FFA or the PPA, okay? So this is the idea of what's going on, but a natural question to wonder, though, is like, well, when you're thinking beyond category selectivity, though, what are all these different voxels and all these other different parts of the brain, all these other little different regions actually involved in representing? And the way that I'm going to try to visual or explain this is with this little toy animal here. Okay. So what's going on in this particular plot is this big panel here is a small patch of cortex from which people have gone all around the cortex and measured neural firings here, for here, blah, blah, blah. And so what we've done now is we've circled all the parts of the brain that significantly respond to at least one of these three little images here, okay? So in this case, whenever you see a red circle, it means that the neurons in those circles get excited when they see this thing, but they don't get excited by this thing, and they don't get excited by that thing, okay? So red circle means it likes this, but when you see no blue, it means it doesn't like this, and no yellow means it doesn't like this. Situation like this, however, means that this little patch right there gets excited by the red one, gets excited by the blue one, and gets excited by the yellow one, okay? So when you compare what's going on in these different circumstances, you can start to see like, oh, different neurons that are nearby one another like different little things. So for example, whenever you see a cluster of these three colors together, you can maybe infer like, oh, this little patch of neurons, it likes this outline of this head. It's a head outline detector because the outline of the head is here, the outline of the head is here, the outline of the head is here. And so what you can see is like, oh, these neurons are going to get excited by all three of these because the head outline is always there. However, for example, you can see like maybe where you see blue and red, you might want to think about those as something like nose detectors or eye detectors or features of the face detectors, because you can see that they get excited by one, which has all the features of the faces, gets excited by two, which has all the different features of the faces. But when you take those features of the faces away between the 2 and 3, you see a difference in activity. Now you see that there is no yellow here, okay? And so the idea is that basically ventral temporal cortex beyond category selective regions is made up of a bunch of these little shape detectors, okay? And it doesn't just have to be a little cat like this. It could be also something kind of like this. Here's another example where you can take this little stuffed animal, deconstruct it to these little circles, deconstruct it even further to those circles. And so what you can see is like, it's like, oh, okay, there's something in these two situations, okay, these two cases here, that get excited by this one and this one and this one. These two locations seem to just always get excited so long as there's this red-yellow. Whereas these situations out here where it's just the red, no, they want something more complex than that. And then you have this thing like it's blue where it's like, well, it gets excited by this. Oh, wait, sorry, it gets excited by this, but not excited by that. Did I just flip that? Sorry, I just flipped the red and the yellow. I think they flipped it. Oh yeah, sorry, I got confused because notice it goes red, blue, yellow here, and then blue, red, yellow there. Sorry about that, everyone. So when you have these red ones out here, the red regions seem to get excited by this little guy, but only the blue regions seem to get excited by that little guy, whereas these ones here get excited by all three of them, okay? And to show you this in the context of real data, here, again, is a big swath of cortex. These lines are obviously the vasculature. And what I've done is just highlighted 3 different sites that are numbered for whatever reason, 2, 5, and 6. And what you can do is you can see that when you show these neurons in picture of a face, site 2 and site 5 get really excited. That's why you see the red. But site 6, a little bit less so. That's why there's only a little bit of red. Whereas when you show it oh gosh, is that an apple or is that a peach? I think that's an apple. When you show it an apple, you can see this one gets a little bit excited, so there's the green there, whereas these sites don't really get excited, so there's no green there. And then just to round it out, when you have the little giraffe, none of these sites seem to get excited by the giraffe, okay? So the idea that I want you to walk away with is just this notion that you have some category selective regions that allow you to represent items like faces, body, scenes, there's also words. And then beyond those category selective regions, you have these finer grained patterns that allow you to represent these more diverse set of objects, like a Discman or duct tape or a wallet or whatever it is, okay? It's thought to sometimes be something like a raisin in oatmeal organization, it was initially thought, because it's like, okay, you have these pieces of bits of fruit, the little raisins that are in this oatmeal that are these finer grained patterns of activity. Now, a question, though, that people have recently gotten very into is this question about, well, is there structure and order beyond these category selective regions for faces, body, scenes, and so forth? Is it really just like chaos, there's no order, there's no rhyme or reason, and so on and so forth? And for a while, people thought the answer to that question was yes. If you were taking this class 10 years ago, what we would have said was like, yeah, you've got category selective regions for things like faces and body scenes. And then out here, it's just madness. It's just a multicolored dreamscape of nonsense and no order and structure. But in the last several years, people have learned that this is in fact not correct. And there is some structure and order beyond these category selective regions. And so let me now walk you through it. So in this particular study, what they did is they first wanted to localize regions that are involved in perceiving faces and scenes. So these yellow regions here are for regions like the FFA, and the blue regions there are for regions like the PPA, which again is a scene selective region you'll read about. And now we're looking at a flat map where you can see the response to faces. So that's what these yellow regions are, and buildings as well, okay? So then what these researchers did was kind of interesting. They not only showed people pictures of faces in buildings, but then they also changed where they would show these items. They would sometimes show them in the center, and they would sometimes show them in the periphery. And what they actually found is that items that were in the center of the display, kind of right where you're staring in the middle of the display, and items that are in the periphery off to the side of where you're looking would activate different parts of the brain as well. So you can now see a color coding of this flat map as well, where you can see stuff that's right in the middle, which they color code as yellow, activates this part of the brain here, activate that part that's a little further out in the periphery, activates the purple, and then parts that are further out still activate, are activated, and you can see color coded by this green region. Now, I don't know about you, flat maps are always a little tricky for me to look at. So now what I'm going to do is I'm going to show you the same pattern of activity but on a more typical brain. And what you could see is something like this, that when you show items in the periphery, it activates these green regions. When you show items in the mid-periphery, it activates these purple regions. And when you show items in at the fovea, it activates these yellow regions, okay? So why is this showing you that it's not just raisins and oatmeal? It's showing you it's not just oatmeal because you can see that that a huge part of the visual system is actually organized by where something lands on your retina. If it's far out in the periphery, it goes to one side of the cortex, this medial surface. If it's right in the middle of your retina, and then like right where your fovea is, it goes to the lateral surface of it, okay? And it actually turns out that there's some relationship between this sort of organization and the relationship, and its relationship to the FFA and the PPA. Where in all this are you gonna put the FFA? You're not gonna put the FFA out in the periphery, you're gonna put it in the center. Because I don't know about you all, but usually when I see someone, I'm gonna look at them with my fovea, I'm gonna foveate on them. PPA, meanwhile, which is a big scene region, is gonna go way out into the periphery. Okay, and so if we actually go back a few slides, we can see that, where we can see that the face regions are in the yellow because they're in the middle of the visual field, whereas the buildings and the scene stuff are out in the periphery, okay? So it's not just a raisins and oatmeal organization. There actually is a structure based on where items are presented relative to where you are. Another reason why people think that it is not just raisins and oatmeal organization is the fact that there's actually also large-scale differences with the type of object that you're looking at. Sorry, my computer just froze for a second. So another organizing principle comes from the dimension that is known as real-world size. So it turns out that if you actually put someone in a scanner and you show them a bunch of objects that are small, literally small in everyday life, you know, these are all things you could pick up with your hand, and you look at how your brain responds to small things versus big things, what you actually find is that there's differential patterns of activation for these real world objects that are big and these real world objects that are small. Okay, why do I keep saying the word real world or the phrase real world? Because notice on the screen, they're effectively the same size, okay? Like this pine cone and this chair and this wheelbarrow and this binder clip are all pretty much the same size on the screen, but you know that they're different sizes in real life. Okay, so it's not perceived size on the display, it's real world size. And what you actually see is that there's a pretty large difference here as well, where small objects will activate one side of the cortex and big objects will activate another side of the cortex. So this is another dimension of organization beyond category selectivity and where something is in space, which is just kind of what something is and specifically how big it is. And then there's just one other dimension besides eccentricity and size, and that's animacy, whether or not the items that you're looking at are animate or just objects. So again, what you can do is you can show people pictures of a bunch of different types of animals, show them pictures of a bunch of different types of objects, compare these to one another, and you can see that, oh, in fact, there's some parts of the brain that get really excited by objects, and there's some parts of the brain that get really excited by animals. And so putting all of these together, you could start to see as literally as much as cognitive neuroscientists know about how visual objects are represented. And it's a couple of main principles. One is through category selective regions. Two is through based on organizing where things are out in the world. Stuff that's in the fovea goes to one part of the brain, stuff that's in the periphery goes to the other. Three is how big things are, whether they're small or large. And then four is whether or not there are images of things that are alive or things that are just objects. And so all these principles have to come together for your brain to be able to recognize the things that you're looking at, okay? So it's not just a one-size-fits-all type of problem. It would have been kind of nifty if it was like, oh, we have a module for a face, a module for a scene, a module for a word, a module for a dolphin, a module for a gorilla, a module for a cup of coffee. But we don't. Instead, we have a few items that are prioritized, faces, bodies, scenes, and words. And then for all these other categories, they have to kind of mix and match and come together through all these different organizing principles for us to recognize all the different types of things that we can recognize in the world. All right, so with that, we're going to call it a day with object recognition. But in the next section, we're going to actually move on to talking about attention, and specifically visual attention.